I'm very pleased to introduce our next speaker, Professor David Seaman from Stanford University. Now I suppose he's going to talk about change point detection in sequential settings, I suppose, right? Because David is a great expert in theory of optimal stopping rules and change point analysis. Please welcome.
Thank you. So it's a great pleasure to be able to return to the Newton Institute, which I first visited in 1998 and haven't visited since then. So this is, as I say, a great pleasure. I'm going to take a rather fast historical tour of change point detection, which began with a fairly narrow mathematical point of view, or fairly narrow applications orientation in the 1950s and 60s. After some rather ingenious mathematical work, it has blossomed out into a field where where the applications are driving modern developments, I think. And so I'd like to describe that history because the applications today are very interesting. They don't seem to bear any relationship to the way the founders of the subject thought about the subject. And yet the methods that are being developed are still building on methods that go back to the 50s and 60s. Now, with regard to applications, I will say that my talk is very idiosyncratic and limited by my own knowledge and the problems I've worked on. So I'm just asking you to take at face value that while I'm going to describe a number of problems in biology, that you can find similar groups of problems in economics and finance and other areas as well. So, as I say, the subject began actually here in England with the work of E.S. Page, who posed the following problem in two papers, 1954 and 1955. Suppose we have a sequence of independent observations up to an unknown parameter k. They are identically distributed with a particular distribution f0, while for larger values they have the distribution f1. These may be completely specified, or there may be unknown parameters. Of course, when we get towards applications, the unknown parameters are a very important aspect of the problem. We can observe the observations sequentially, and in fact, this was Page's original viewpoint in his first paper. And our desire is to detect the change point k as soon as possible after it occurs, while rarely claiming a detection before it occurs. In other words, the goal is to define a stopping rule which with very small probability satisfies, it takes on a value less than k, and with very large probability takes on a value only slightly larger than k. There's also the possibility of a fixed number of observations, which has become much more prominent in current applications. In that case, we might, for example, want to test the null hypothesis that f0 is equal to f1, but what I will point out and emphasize throughout is the essential feature of the problem is that k is undefined when f0 is equal to f1, and so you could easily well specify the null hypothesis as k greater than or equal to m. And in fact, there's a whole class of other problems that have this same essential feature, which makes them irregular from a statistical point of view. Therefore, for example, there's no automatic equivalence, even asymptotically, of Bayes and likelihood procedures. They may be the same or are asymptotically equivalent, or they may not be. And each problem has to be examined more or less on its own merits, because there's no overriding theory, but there is, I think, an overriding set of approaches to the problem. Now here's Page's solution to the problem that he posed for sequential detection. He suggested that we take the cumulative sum, which led to the name Q-sum test, of some scores, z associated with xi, and then we stop the process and declare that we've seen a change as soon as this random variable exceeds the threshold b. Page did not emphasize, I think it was Lorden's observation to say that this is the dominant score, and there's no sense considering anything else when f0 and f1 are completely specified. Of course, if they're unknown parameters, there is reason for considering something where we try to estimate parameters. But Page did not say that, at least not very emphatically. But if we assume that, then you can see the rationale very clearly behind Page's procedure. If we have independent identically distributed observations and they initially have the distribution f0, then this random variable has negative mean. So we have a random walk which, as time proceeds, tends to go in a negative direction. And then at the time k, where the distribution changes to f1, it tends to go in a positive direction. So subject to fluctuations, at any time, if we want to ask, have we passed the change point, we should compare our current value to how low it was before at some earlier time. And if that distance is large enough, say there's the threshold b that's written there, then we stop and declare we've seen a change in this part of the path when each new observation is in fact likely to be the minimum value. There's no evidence whatsoever for a change. So that's the basic logic that drives a large number of procedures. However, the minute we introduce unknown parameters, things become somewhat more confusing. Barnard in 1959 suggested, suppose we do this for a normal random variable delta, there's the likelihood ratio if delta were known, but if we maximize out delta by putting in the maximum likelihood estimator, then we get a much more complicated procedure because it now involves this nonlinear function. And in fact, while Page actually analyzed his rule with a fair amount of precision, and other people jumped into the fray and posed alternatives, alternative analyses after that, it was another 30 years before there was a coherent analysis of Barnard's proposal. But maybe I should emphasize, it involves the same idea. That is, you look at the current number of observations and you look backwards and you say, was there an earlier time when the mean value of the random walk between the earlier time and the present time seems to be not zero, as measured here by just the standard z statistic. Of course, there's a multitude of multiple comparisons because we're stopping with regard to t, that involves multiple comparisons, and we're maximizing with respect to k, which also involves multiple comparisons. And that really is the charm of the theoretical aspects of the problem, is dealing with these multiple comparisons, which arise naturally but in a well-structured way. Shuryaev, so the center of gravity switched from England in the early 60s to what we used to call the Soviet Union. Shuryaev, who I believe was Kolmogorov's last student, and I guess arguably his best student, considered the case of a completely specified f0 and f1, but he made k a random variable and formulated a Bayesian version of the problem. His version was if you claim a detection at time n, you incur a loss. Well, if n comes before k, you lose some penalty because you've stopped too soon. If n comes after k, you measure your penalty by the length of time it takes you to recognize that a change has occurred. Giving k a random distribution, he arrived at the following Bayesian type rule. You look at now a sum of likelihoods. Well, I should say this is an asymptotic formulation of his problem when we consider a geometric prior with a small value of p. That is, it's unlikely that a change occurs early in the process. It only changes, there's a change that occurs only because we go on sampling for a long period of time. Then where Page considered, so to speak, a maximum functional, that is, here you see it more clearly, the maximum of the process up to time t. In the Bayesian formulation, we consider a sum of likelihood ratios, and then an approximation to Shuraiev's stopping rule is given by this thing that I've called N1. Now, the fundamental properties are that for both of these stopping rules, the expected value when there is no change, that is, if we imagine that we can actually go on sampling forever because no change has ever occurred, then the expected wait time until we stop, which means making a mistake, is proportional to the exponential of the threshold. I described the threshold once in terms of little b, once in terms of big B, because once I was in Page's procedure, I was thinking about a logarithmic scale. In Shuraiev's procedure, I was thinking about the original likelihood scale. So the expected length until we make a mistake, if there is a mistake to be made here, can be very, very long, while the expected length of time, the so-called expected delay, is relatively short. That is a logarithmic in this so-called average run length time. It's a little bit difficult to identify these constants C, but the proving that this is the right order of magnitude of the problems is quite straightforward and was done by both Page and Shuraiev early in the process, and then refined by others. Now, I want to mention now a contribution of Davies, 2 papers in Biometrica, pointed out that there's a class of problems having the same essential feature, but where we don't have this kind of jump discontinuity. The problem is, he referred it as a problem of the existence of a nuisance parameter that's only present under the alternative hypothesis. So remember that I said that if we think of k, the time of the change point, as being the essential parameter, then the difference between f0 and f1, that parameter only exists when k is less than the sample size. It doesn't exist otherwise. So this has the same essential irregularity, even though technically it's a different kind of problem. The log likelihood might be smooth or piecewise smooth. And in fact, although Davies pointed out this connection with change point problems, there was several papers, a couple of very famous papers in 1939 dealing with a version of this problem. This is just to say a special case of the problem that Hotelling posed. We have a regression problem, but where the regression function depends on non-linearly on an unknown parameter. Here's a simple example that's gotten a certain amount of attention in the literature. We have a change point in the slope. That is, we have a regression curve that is just flat at the level alpha, and then it takes off like that. So, but the essential feature is that if beta is equal to 0, theta is undefined. That's the essential feature of this problem. Hotelling wrote about this problem in 1939. Hermann Weyl, in a very famous paper, begins by saying, a few months ago, Hotelling gave an address at the Princeton Mathematical Society, and he posed the following question, which I will now solve. Hotelling's problem involved a one-dimensional parameter theta. Weyl's involved a multi-dimensional parameter theta. And the difference in the geometry involved is enormous. Hotelling did what most of us would do with, you know, what you learn in one-dimensional calculus about curves and arc length. Weil's paper is a very famous one in differential geometry because it became the basis for the first quite general version of the what's it called? The Gauss. No, the theorem of Gauss about curvature. The Gauss-Bonnet theorem. Thank you. So this became the basis for Allendorfer's generalization of the Gauss-Bonnet theorem, but it was promoted by Hotelling in discussing the problem of statistics. Now Davies actually used Rice's formula. Davies actually used Rice's formula, which I won't try to tell you what that is if you don't, but it's a commonly used formula in applied probability, to evaluate significance levels. And I will mention this because these three papers were published in the same year in the same journal, the American Journal of Mathematics. Now, there's good reason for Weil's paper to be published next to Hotelling's. He was motivated by Hotelling. Rice was employed by Bell Telephone's laboratories and was writing about problems of communication theory and electrical engineering. And the connection here between Rice and Hotelling and Weil was not recognized, I think, for another 30 or 40 years, but it's there. So there's this very interesting early history. Much more recently, Worsley has developed a model for brain scans which builds basically on the model of Davies. That is, we imagine that we have a signaling process that we don't observe directly. We observe its convolution with a kernel, which may have a scale factor in it or may not, but it has a location in any case. The kernel can be assumed to be standardized, and that's a convenient standardization. The hypothesis of no signal, where we're looking for some point of activity within the brain, the hypothesis of no signal is that dx is just Gaussian white noise. The signal is that dx is given by a nonlinear regression. That is, some kind of regression function about the signaling, the point of the signal tau, that has a scale to it and an amplitude to it. And now, if we know what we're doing, which we don't in this problem because there's no particularly good shape for this signal to have, we would match f to f0. F is under our control, basically. But, and sigma to sigma 0. But we can't do that, so, but in any case, we'll take and look for a detection, a peak in the signal here, which is very similar to the peak that you get if you imagine that you have this segment of data and you ask yourself, what's the maximum difference you can find between some point here and some earlier point. But this is a slightly different problem. It fits into Davies' scheme, and again it has the same essential features as a change point problem, namely that if the parameter xc is 0, then the parameter tau is undefined. If there's no signal, there's no change point. Conversely, if there's no change point, there's no signal. Worsley applied geometric methods going back to the Hotelling-Weil kind of argument, but these had been developed by Adler in his book on Gaussian processes and fields in about 1980. So the direct application that Worsley used was to Adler. To estimate significance thresholds, again, there's a multiple comparisons problem by virtue of looking over the whole brain at different scales to see if we can find a place where there's a little bump in the data. Now, this was we can imagine putting both of these problems together, and I'll mention this just briefly because it's a pretty complicated problem, but to have sequential detection of a structured image, if you've sort of created some intuition about thinking about brain activity, you might think that a stimulus is presented and then a signal appears at some later time, and that's what we would like to detect. That's what represents a change point. So an image is a finite collection of pixels. In fact, maybe we go to the picture and it makes more sense. So we have a finite collection of pixels. Here just imagined as to be a square in the rectangle in the plane. We have an image, which is a signal, somehow a little bump that we could see if there were no noise. When there is noise, with a single observation, we don't see anything. Even with 20 observations, we don't really see the pattern. With 100, we see the pattern and know without any statistics. This signal can be detected in about 8 or 9 observations, so much before a pattern appears to the naked eye. The formalities, we don't have to go back to the preceding, well, for just a moment. So the shape of the signal is given by some real valued function g, defined on the set of pixels. Its intensity is a positive number xc, as in the notation I used for the brain scan. And then we make an observation at the point t that depends on the signal that's present at the pixel j. And we search around until we try to maximize that to detect where the signal is located. We do this by forming a likelihood ratio statistic. There's the statistic for a single observation. And then if we try to jazz this up using what we've learned from Page and Shuryaev and Worsley, then we can parameterize our signal by the amplitude and the location. We form a likelihood ratio, which involves the sum of these inner products. And now we want to detect the signal, which we can do either by, well, I haven't told you yet. So we'll make a constraint on the false positive just as before, and we want to detect a signal we want to detect the signal as soon as possible after it occurs. And now we're going to change the criterion a little bit. Instead of just looking at the expected delay time, we're going to look at the expected Kullback-Leibler information, because this allows us to do something uniformly in theta. If we look at the expected delay time, then we get tied up in various technical problems. But based on what we've seen before, we expect that we can make this rather large compared to this. That is, this should be logarithmic in that. And in fact, we can do it. I've written here some formal description, but I think because I'd like to spend more time on the later part of the talk, I'm just going to say there it is, and now we go on. Now I want to move to trying to describe a number of biological problems which have the same basic structure. We observe a process. This is designed to be to simulate thinking about linkage analysis, which is the goal of which is to find the genes that are contributing to a certain phenotype. You should imagine this goes on forever, and most of the time it's just random noise centered around 0. A signal is a point tau where we have a peak. And in linkage analysis, the peak will have this sort of double exponential decay shape. And so we make a very large number of observations in the hundreds or tens of thousands, and we look for high points relative somehow to the number of observations. So again, we have the multiple comparisons problem. Again, we have the problem that if xc is 0, tau is undefined. Again, it's a change point in the in the form of a cusp. You could think of it as a change point in the slope if you like. But it has the same basic structure. I will not spend much time on this problem because I would like, so this is just a summary of what I said. In linkage and association analysis, we look for large values of this process as we pass along the genome after standardizing so that under the null hypothesis, we have a standard normal distribution. And the models can get quite complicated because there may be several of these peaks. They may interact with each other. They may interact with the environment. So, so this is certainly work in progress, although the principles are well understood, I think. The details of doing this in particular cases are widely practiced by large numbers of biomedical scientists around the world with varying degrees of success. Another example in the biological vein is this paper by Denbo, Carlin, and Kawabata in working on DNA protein sequence analysis. We attach a score to each amino acid in a protein. On average, the scores are negative, as in the case of the log likelihood ratio. In fact, they basically suggest that one should use log likelihood ratios. What we're interested in is finding a high scoring segment. The scores, the two examples that are mentioned in this paper are hydrophobicity, that is the extent to which the amino acid does not want to be dissolved, tries to avoid water, because this gives you some idea when the protein folds up in a ball, what's on the outside and what's on the inside. The molecules on the outside have to be willing to be in contact with water. Molecules on the inside frequently do not want contact with water because they'll just dissolve. So, so measures of hydrophobicity are one example. Measures of electrical charge are another example. So we want to know, in this long sequence of amino acids, most of which have negative score, is there some segment where there's a positive score? So unlike each time I go over there, I look for the chalk, I pick it up, I write something on the board, then I bring it over here and I drop it, even on the floor. So in this case, we're looking for a pattern that looks like this. That is, there's a high scoring segment somewhere in this thing. Or you can imagine that if instead of saying, we've reached a turning point, we want to detect this, we imagine that we let the data go on and then it reverts back to its original level and then goes down again. So as you might guess, the statistic that was proposed to study this problem is actually very similar to what Page had talked about. But when they started looking around in the literature for somebody who might have studied this problem elsewhere, they were led to Eigelhart, who was interested in queuing theory, because this statistic also represents the maximum waiting time up to time, capital T, the maximum waiting time anybody experiences in a single server queue. That was Eigelhart's version of the problem. There's Page's version of the problem. And now we have the Denbo, Carlin, Zatuni version of the problem. But by that time, it was sufficiently well studied for other reasons that they're just able to quote Eigelhart to give a p-value for this statistic. There's a much more complicated version of this of this problem when we do DNA or protein sequence alignment. when we try to align two proteins to see how similar they are. Dembo Carlin and Zaytouni wrote about that in 2 papers, and Sigmund and Yakir, with a mistake in the first version, extended their results to the case where you allow gaps in the comparison. I won't try to describe the whole problem, but this is a much harder version of this problem. And as I say, in a certain sense, the question that's being addressed is a question that Page could have asked, but it wasn't one that he knew how to solve, so he didn't ask it. He formulated the problem differently in the time-honored technique of changing the question if you can't give the answer. Now another problem that appeared fairly recently and has probably got more attention than any of these other problems except for the DNA sequence alignment is the problem of copy number variation. In an ordinary human genome, you inherit 1 copy of a little snippet of DNA from your mother and 1 copy from your father. But it was observed rather early on by people with powerful microscopes that at least in cancer cells, you can have multiple copies of DNA, or you can have pieces of DNA that are missing. And that refers to copy number. So you can have 0 copy numbers of a certain segment of DNA that should be present. You can have 1 or 2. 2 would be normal. You could have 3, 4, 5, or 6. And since one can observe this in cancer cells, the question naturally arises, could this possibly be a cause of the cancer, or is it a symptom of the cancer? What relation does it have to the cancer? So trying to find copy number variations has become a fairly big business. This was the original paper that piqued my interest in the problem. We assume we have observations that are independent and normally distributed with unit variance and with mean values equal to mu sub I, but which are subject to a whole sequence of change points. So tau 0 is 0, but then at tau 1, mu sub I changes. So the parameters tau sub I and mu sub I in general are all unknown. Our goal is to decide the correct value of m and the change points tau 1 to tau m. Now, we can do some analytic analysis if we assume that capital T is very large and that m is much smaller than capital T, while these increments are sufficiently large that mu I can be estimated accurately. Otherwise, we'll get into serious problems. Of course, the minute we prove a mathematical theorem of this kind, we now try to apply the method even when the conditions aren't satisfied. Sometimes we're successful, other times not. I should say, I'm going to talk about several other variations on this kind of problem. And I should say that this is a simplified description. And one of the issues that you have to deal with is that there's drifting. There's sort of a slow oscillatory drift in the signal. And you will over-detect change points if you don't do something about that slow oscillatory drift. It has something to do with the CG content of the DNA. And some people have suggested that should be used as a variable and take the residuals from a regression on the CG content. We've used a different purely data analytic technique, and at the end, we think this model is quite reasonable, but the data have been massaged quite a bit. And I won't try to describe that. So here are a couple of variations. There's a baseline value, say 0, or the log to the base 2 of 2. That is, 2 is the normal value. So there's a baseline value such that you return to the baseline. That is, you have departures from what's standard, say two copies, but the departures are actually pretty rare, and then you come back to the baseline, then later on there's another departure. This can happen because there are also inherited copy number changes. I'll show you some data later on, and you'll see that the inherited copy number changes are relatively rare and relatively short. They're not easy to detect. But population geneticists are interested in them because if you and I have the same inherited copy number variation, we're probably related. Maybe 100 years ago, 500 years ago, who knows? But it's another way of building a coalescent tree relating you to me. So there also can be n sequences of observations. That is, instead of having one DNA sequence, we have n sequences, and we look for common changes. This is what I was just mentioning with the changes due to inherited values, because common changes will indicate inherited values. So we'll just build a model where we assume that the changes occur at the same place. And there's a subset of the sequences which have that change. As I said, these intervals tend to be very short, and therefore they're not easy to see. But if we have a number of sequences of data, then we can see those intervals, as I'll show you in a moment. In the gene mapping experience that I mentioned before and passed over rather quickly, there actually are typically a number of genetic loci unknown, and there can be kinds of interactions between them with environment. So again, the problem can be much more complicated than what I've described so far. And I'll just deal with one of those complications, namely this one. So here's an example of some data that was used at Stanford basically to debug a procedure. I'm not going to explain it in detail except to say this is the this is the raw data on a particular scale. I'm going to show you the same raw data later, and for some reason the computer chose a different scale and it looks different. But you'll see the similarity. You can basically see where the changes occur. There's an interval where there's a loss of DNA. That might be an interval where there's a gain of DNA. It's sort of picked out down here as an interval where there's a gain of DNA. Here there are 61 sequences, and some of these sequences involve replicates. That's why this line has a certain amount of thickness to it, because there are replicates in there. Again, I don't want to try to describe the data in detail, or we lose the, I won't be able to finish the talk. But this is what the inherited copy number changes look like. They're relatively few. They can be rather short, and well, probably we can see most of them, although it's not so clear that what we would decide about that if we didn't process the data. So what do I mean by processing the data? Well, here's the statistic proposed by Olshan and Venkatraman for a single sequence. It's basically just the likelihood ratio statistic. But it's a likelihood ratio statistic where we don't know where the changes occur. So here we're imagining that there's only one set of changes. That is, there's maybe a high level or a low level. Begins at S, ends at T. We don't know where S and T are. So we maximize over a possible S and T, but we just use the likelihood ratio statistic, assuming the data are normally distributed as I specified. Then we have to deal with multiple comparisons, and I will not try to do that in this talk. When we have a model for aligned CNV, that is, we have a two-dimensional array, there's one other, the problem is sort of spelled out here. Well, remember I said there's only a subset of the profiles which exhibit any particular change. So, where's my chalk? So you can imagine that what we have is something, a noisy pattern. a noisy pattern with a little change in it, another noisy pattern, another noisy pattern, another noisy pattern with a change in it that's assumed to be aligned with this change. And of course, we're not really limited to a single change, but the model, I'm going to consider the model of a single set of changes, but with the understanding that this number here, which is 2, is small compared to the total number of sequences. And so I don't want to consider simply a multivariate likelihood ratio statistic, although that's possible and we did it for a while. Rather, we introduce a pseudo-Bayesian type of parameter. You can think of it as just a way of enforcing sparsity, or you can try to interpret it as a probability denoting the fraction of the profiles that have a change between S and T. And here the change is delta sub I for the ith profile. The changes can be different, and that's why I drew one of them going up and one of them going down. They don't have to be the same in the same direction. Maximize out delta, so we get something that involves what if P0 were 1 would just be the likelihood ratio statistic, the multivariate likelihood ratio statistic. Now, however, this enforces a certain amount of sparsity, but it complicates the analysis. So in the end, we analyze sort of general statistics of this form, and we're able to derive some p-values for them. But I omitted that slide because I want to pass on to one other subject. So what I've said so far builds quite directly, although technically can become very complicated, but conceptually builds quite directly on the framework of Page, Shuryaev, and others from the beginnings of the subject. Another approach involves a model selection approach. Now, there are many model selection approaches, and a number of them have been suggested. I'm going to talk about only one because it's the one that I have tried to understand. I won't say it's the one I do understand, but it's the one I've tried to understand. And that's the BIC criterion, which was actually employed in a change point problem fairly early. But what was not observed is that the standard derivation, let me back up. BIC criterion was proposed originally by Schwarz, who said, what I would like to do is make a Bayesian problem where I have a bunch of different models. I'll look at the data. I'll calculate the posterior probability given the data for the ith model, and I'll choose the model that has the largest posterior probability. Since that's a difficult problem, I will compute an approximation to the posterior probability. Now, that approximation involves certain assumptions. And here's the classical formulation. Excuse me. We have a number of regular parametric models. Now, regular means in the sense that maximum likelihood is what you expect it to be, and the Bayes estimate is going to be roughly equivalent to the maximum likelihood estimate, and so forth. In particular, you can differentiate the log likelihood twice, and when we compute a posterior probability, assuming a smooth prior density, we use the Taylor series approximation, and we arrive at this criterion. We maximize over k, the maximized likelihood for the kth model, with a certain, what has come to be interpreted as a penalty. Now, once you interpret this as a penalty, you can apply this to any problem. It doesn't have to be one that satisfies the assumptions leading to this formula. And that's what has happened, with the usual conclusion that it's not a very good procedure, but the wrong penalty has been used. So It's not a penalty that corresponds to a Bayes analysis of the problem. There's another technical assumption that the number of models that are considered is bounded. If you consider an unbounded class of models, you again get a different criterion. And if you imagine that you have a very large sample size, it's also natural to consider that you might have a very large number of models. Or alternatively, if you think in terms of a change point problem, where we have one change point followed eventually by another change point followed by another, large sample size means we have a long sequence of data, and why shouldn't there be a large number of change points? Why should we impose an assumption that that number is bounded? For a single sequence, Nancy Zhang, a student of mine at that time in her PhD thesis, derived this criterion, which still involves a maximized likelihood. It still involves a parameter, a penalized value of the number of the log of the number of parameter, or the number of observations, but involves this other condition, which measures the accuracy which with we can estimate the mean values in between the different mu sub i's. For reasons that I'll not try to describe in this talk, we became a little bit dissatisfied with this and thought of the problem and asked ourselves, why should M be assumed to be of some fixed size when the data we think of as being very long? Should we also allow M to be large? Well, I'm sorry. Let me make some general comments first, I guess. So one point that Schwartz made was that the classical BIC procedure does not depend on the specific prior distribution. So you can pretend to be a Bayesian and then not a Bayesian at the same time. Of course, modern Bayesians think this is a defect. I think it's a strength. Although the procedure would depend on the specific prior distribution, if you carried out the expansion of the Bayes factor to terms that are of a constant order of magnitude, or if the number of models is allowed to increase indefinitely with the sample size. There's a very nice paper by Berger, Ghosh, Mukhopadhyay, which points this out and gives a nice discussion of this problem, where the problem is regular in all of the respects, but when n becomes infinitely large, the sample size, you also allow k to tend to infinity. And if you do that, you find that you have to, your prior does make a difference. And we modified their prior a little bit, and we actually, since now the procedure will depend on the prior, we consider two different priors. There's a scale factor, which we can estimate by empirical Bayes, because remember, we are considering different mean values, so this tells us what the fluctuations in the mean values are. And we either use the covariance or the identity function. We assume that omega is to make this Bayes problem, it has to have a smooth density. It's positive and bounded on zero infinity. And we take the taus to be order statistics from uniform distribution, the change points, the order statistics from uniform distribution, although that's not critical. And then we get different BIC criteria. Here's the one based on the first prior, here's one based on the second prior. They look somewhat different if you look at them carefully, which I don't propose to do. Numerically, they are very similar, so it probably is not terribly important that, strictly speaking, they depend on the prior. There's this amusing feature of the problems that in Page's formulation, which is maximum likelihood, you end up looking at this functional Brownian motion, assuming you scale things in the right way. You end up looking at this functional Brownian motion until you're sick and tired of it. In Shuriya's formulation of the problem, which involves a prior distribution, and therefore summing over likelihoods, you end up looking at this functional of the distribution till you're sick and tired of it. In this formulation, because we're sort of trying to have our cake and eat it too, that is, we're giving a maximum likelihood type of analysis of a Bayesian formulated problem, we end up with both these functionals. They don't play a terribly important role. We can compute these, and they don't sort of determine the story, but they make it interesting mathematically to find these different functionals popping up. We can also do this for multiple sequences. I won't go into the details, but now I'll show you a couple of examples. Oh, here's the formula for multiple sequences. Again, too complicated to try to digest, but you see we have to estimate the fraction of the sequences which actually show a change point at any place. That becomes a new parameter that we have to estimate, which again we do by an empirical Bayes argument. Here are the data that I showed you a minute ago, and as you probably have forgotten by now, but the computer chose to plot the raw data on the scale of -2 to 2, and now they're plotted on the scale of -1 to 1. So this looks like it's a lot noisier. It's the same data, just different plot. And the BIC criterion sort of picks out, maybe with a little more emphasis, again because of the different scale, the same regions, although this region as a darker region than that one does not appear in the original analysis. But now, when this really becomes interesting is when we have cancer data, because cancer data are much noisier, and there are many more change points, and some of them are very long. As I said, some of these regions you can see under a microscope. You don't need statistical analysis to see them. And so, and whereas the method that I discussed earlier, where we do repeated hypothesis testing, it only really makes sense when you think there may be no change points. Then it makes sense to ask yourself, well, what's the evidence that there really is a change point here? Here, there are change points all over the place, and somehow model selection appears to be a little more appropriate approach to the problem. Here are references that I mentioned during the course of the talk. They're by no means complete. I quit when I got to the bottom of the slide. And as I say, particularly with regard to the model selection point of view, there are many different approaches, and we will hear at least, during this workshop, we'll hear at least one other approach, maybe others. And so I shouldn't, as I say, this is a historical idiosyncratic survey, not a systematic survey. And I hope that, thank you for your attention, and please fire away on the questions if you have any. Thank you.
About the applications for copy number variations, you mentioned that they're also important because of sequencing. Now the new sequencing data, and so I suppose the difference is that now you have a lot. I mean, you have very long signals, the millions probably. So how does this method scale? I mean, can we apply all these tests? Do you need to reduce resolution or something?
We are currently working on a method to use sequence data. And I think the main, conceptually, it's a very similar problem. You are working on a completely different scale, whereas here, I didn't say it, but an observation represents maybe 1,000 or 10,000 DNA letters. There you can actually detect omissions of in principle, omissions of a single letter. But the data can't be thought of as being normal, in my opinion. We're working on a problem involving Poisson modeling of the data with a similar point of view, and it's work in progress. It's not complete. I think I'm going to give a talk a couple of places on that subject, but here I was asked to give a review, so I reviewed the stuff that I think is to some extent settled.
Well, any further questions?
Oh, well, I'd like to add up, maybe it's not a question, but that there are a lot more applications that David mentioned for one problem. Can you go back to the slide where you consider this structured or unstructured, doesn't matter, changes with the multiple data streams? Can you introduce the statistics?
Just tell me when to stop.
Okay, so that's the slide that you considered with the unstructured changes with this P naught.
Yes.
Remember? Maybe go back.
It's this one.
Yeah, that's one. This is a very important problem in many applications. One application is detection of targets. And I worked personally with this. Say you have multiple objects and you have the sequence of images. Images may be very cluttered and noisy. And you have an unknown number of targets that actually appear at unknown points in time and disappear too. So you have a change point problem. You don't know how many targets. And you have a really multi-channel system because you don't know where the targets are located, and you don't want to make this image as a one image of. So you want to extract multiple data streams, and you don't know how many. But you know for sure that say you expect that there will be not more than 20 or 30 targets, but the number of channels may be 1,000. So you have small percentage, and you know this P naught. And you don't know how many, and you don't know where this segment is. And this statistic will, if it's Gaussian distribution, which is usually not, but very often you can approximate it in this sense. And this statistic, I believe, will be very efficient in this problem. That's one application, in addition to what David considered, where this kind of approach is extremely important. And I don't think that anybody used it. We're just starting to look into it. Okay.
Thank you for those comments. In fact, now
there are many more, of course. I just
Right.
mentioned what I'm working with.
Yeah, let me comment for a moment on your comments, which is to say that I chose one of two possible subjects back here. Here I imagined we have an image, that is, we have a bunch of pixels that are giving a signal, but I imagine that there's some structure to them. There's correlation between the signal that's given here and the signal that's given there. You could also imagine that there are just random points out here that are somehow giving signals, no relation whatsoever. And the model that I discussed that was mentioned just a moment ago is the fixed sample version of that problem, because here these DNA sequences are assumed to be completely independent. I can't say that because I got a signal from process number 10, therefore I'm going to get one from number 11, perhaps attenuated somewhat or something like that. Now there's a convex hull of two problems, one with a well-defined structure, one with no structure whatsoever. There've got to be lots of problems where things that are closer are more likely to show the same behavior than things that are farther away, but there's no reason that that should be measured by a nice metric like a spherical distance or anything like that. Or these things themselves might, the spherical distance might be fine, but the detectors are not equally spaced. And so again, if they're too far apart, you might as well treat them as independent. If they're close together, a signal appearing here is likely to appear here as well. And can you make a model that somehow lies between these two extremes? We've just dealt with what I think are sort of the easy cases of extremes where we pretend we know a model or that there is no model relating the different sequences. And those are problems that are not trivial, but are, I think, conceptually easier because we haven't tried to formulate what we mean by related targets. So thank you very much for that comment. There's a lot to be said about it.
Right, it seems that we should stop. It's 3 o'clock exactly. So
Yes.
please join me to thank David.
Thank you.
It's very nice.